Hello. Hello. Jay, Dr. Jay, sir, whether you have
Good evening,
joined?
sir.
Good evening. Good evening. Good evening. Very good.
Very good.
Yes, yes, yes.
So, our speaker is here, Dr.
Okay.
Shital Thacker.
Shital, please, Shital.
Yes, sir.
I'm very happy today that you are ready to present your work.
Yes, sir. Same
And,
here.
and today's lecture will be recorded. So, second speaker, who is the second speaker?
Dr. Kumari. I will call her and ask her. Maybe
Okay.
she is not here.
Yes, Shital is from which area, which place?
She is from Ahmedabad only, Gujarat.
Very nice, very nice. Okay. Okay, then let us wait for 2-3 minutes and we'll start.
Sure.
Okay. Yes.
Good evening, everybody. I think our second speaker has also joined us.
Very good. Second speaker from which place?
She is from South India.
Okay. From which part?
I think that we will soon see.
Okay. Second speaker, what is the name?
Dr. Kumari, you can unmute yourself.
Yeah. Dr. Kumari, please.
Yes, sir. Good evening, sir.
Good evening. You are from?
Sir, I'm from Rohtak, Haryana.
Okay.
Very good. Kumari,
Yes, sir.
today I really appreciate you
Thank you, sir.
because you are dared to present your research work. Very nice.
Thank you, sir.
Okay. Hello, Jay. You can start.
Sure. Good evening, everybody. Today is our second Sunday of this PhD webinar series that we are working on. And it is our absolute pleasure to continue this series. And I think this shall continue forever in the interest of subject. And in interest of time, I would say I would just like to welcome today's speaker, Dr. Shital Thacker. But before that, I would just like to add that our Gujarat State President, Dr. Meena Trivedi, she is walking something today, so she could not be here, but she sent her warm regards. So now I would just quickly like to welcome our today's speaker, Dr. Shital Thacker. Actually, Dr. Shital is currently working as a lecturer in Department of Microbiology, Institute of Science, Silver Oak, Ahmedabad, Gujarat. Before that, she has worked as a senior research fellow for a DBT project titled Non-Acidic Green Biohydro Technology for Valuable Metal Extraction on Mobile Phone E-Waste PCBs. Also, she has worked as a GRF on the same project. Earlier than that, she has worked as a junior scientist for Kutch Crop Services. And Dr. Shital has done her PhD from Gujarat University. She has also done an MPhil from Gujarat University, and she has done her BSc and MSc from Kutch Bhuj. And other than that, Dr. Thacker has published more than a dozen national and international research papers. And yeah, I would like to welcome Dr. Shital.
Thank you, sir, for introducing me. A very good evening to everyone. I'm feeling honored to present my research work at Microbiology Society India. For that, I would like to thank Arvind Deshmukh sir, Professor Minnal Trivedi ma'am, and Joy sir. Uh, Let me share my presentation. Mm, Sir, you have to enable the screen sharing for me.
Sure, sure. Just a moment.
What do you want?
Are you able to share now?
No.
Okay, so I think only Deshmukh sir can do that, maybe.
Yes. Only host can disable.
Yes, yes.
Is there any problem to you for sharing?
Yes,
Yes.
please make her co-host, sir.
Yes, yes, yes. 1 minute.
Sure.
Shital, huh?
Yes.
Yes, sir.
One minute, please. I'm trying.
Sure.
I'm not getting that option.
I think she is being a co-host now.
She's
She's
co-host.
Yeah.
Yes, yes, sir.
Now you should be able to do it, I guess. Okay,
Is it
perfect.
visible?
Yes, yes, very much visible.
Yes, it is visible.
Okay. So I'm Dr. Shital Thacker and especially mobile phone. And for that, our lifestyle has been changed from agricultural to industrial. And this change leads to the production of more and more electronic appliances. eh, Electronic appliances. And that's why the electronic waste generation has been increased. If we can see the first figure, we can see the graph of electronic waste generation by a different year, and we can see in 2019 53.6 million metric tons of electronic waste has been generated. It is seen that that much of electronic waste can form around 5,000 Eiffel Towers. In second figure, we can see the different countries who has generated the most electronic waste, and India is the fourth largest country which is generating this much of uh, 2 metric tons of electronic waste. In third figure, we can see the electronic waste generation in different cities of India, and Mumbai is the largest electronic waste generating city, but Ahmedabad is also at the top 10 electronic waste generating city. It is standing at the 7th position. Why did I choose mobile PCBs? Because mobile PCBs has higher content of precious metal than other electronic waste like computer or TV or fridge. Other than that, mobile phone has more than 40 elements, and the average metal to mass ratio of in mobile PCBs is higher than other computer PCBs and other PCBs. That's why I choose the mobile PCBs for my work. There are different methods available for the electronic waste recycling, like we can reuse it. If we reuse the whatever the electronic appliances, it is cutting around 30% of carbon oxide from the recycling or treatment method. But some appliances cannot be used for the reuse option. So it is essential to treat it. So formally, only 15% of electronic waste is treating in India and other countries. Other than that, electronic waste are just ending up at landfill or informal treatment, like acid wash and incineration. In landfill, 40% of electronic waste is end up at the landfill with the municipal waste, and it is seen that hazardous metals is incorporating due to this treatment, and finally it end up to the groundwater and finally to the food chain. In acid wash, different hazardous acids are used, and it is also generating the effluent, and it is ending up the pollution to the environment. In incineration, it emits the toxic pollutant and like dioxins, furans, PCBs, and PHs. So it is also generating the pollution to the environment. Finally, recycling can be done by three different methods, like hydrometallurgy. In hydrometallurgy, different acids and chemicals are using for the treatment method. And in pyrometallurgy, heat treatment is used for the recycling. But both of these methods are used at a higher temperature and higher pressure. And also it is creating the issues for the environment. We have the option of biohydrometallurgy, in which microorganisms could be used for the treatment method. Biohydrometallurgy method is used at the ambient temperature and ambient pressure, so it is eco-friendly method. Not other than that, the final effluent is also less than hydrometallurgy and pyrometallurgy method, because it is a closed loop process. So we can use the final effluent and we can reuse it. So the effluent generation is also lesser than other methods. There are two sides of electronic waste. One is the brighter side. As I told, that mobile phones and other electronic waste has a higher metal content like gold, copper, aluminum, iron, platinum, silver, and palladium. The content of gold in electronic waste or mobile phones is less, but the value of gold is higher. It is seen that in mobile phones, 1 ton of mobile phones, 20 to 30 times higher gold present than the ores. So if we only recycle the electronic waste generated in year 2019, we can generate around 9,000 million euros of gold from that. Copper content is the highest in electronic waste, because the most of metal content, around 30% of metal content is copper. So the value of copper is also higher. It is around 10,000 million euros. So if we recycle these metals, we can generate a secondary ore to produce the metals, and it can be reused for the manufacturing companies. But there is also a darker side of electronic waste, because electronic waste has many toxic metals like arsenic, mercury, cadmium, lead, and if copper is in a higher content, it is also toxic. So these all metals will lead to the damage our health, environment, soil, water, etc. So significance of this study was to recycle, to develop any technology to recycle this metal, so that the manufacturing companies, electronic companies, which are already using 50% of the produced metal from the ores, they can use these electronic waste as a secondary ore, and could be use these metals for the production of the new electronic appliances. So the research was designed to develop a process which is environmentally and economically sustainable. So there were 6 steps for this study. First, the mobile PCBs preparation was done, and the metal analysis of that mobile PCBs was done. After that, the bioleaching of the base metals, mainly copper bioleaching was done using the iron oxidizing bacteria. Third step was the bioleaching of the precious metals, gold and silver was done by using the cyanogenic bacteria. And after that, the lab scale bioreactor study was done, which was previously done with the iron oxidizing bacteria and cyanogenic bacteria. After that, the copper recovery was done using cementation method. And then the final leachate, which was, stripping solution which left was recycled to regenerate the metabolites for the leaching process.
Hello.
So, hello,
Ah,
hello, am I audible? So in the first step, metal analysis and preparation of the mobile PCBs was done. The mobile phones was purchased from the scrap market and different mobile shops from Ahmedabad. And I separated these mobile phones according to the type of mobile phones like basic phones and smartphones. And I did the metal analysis of these both phones. And we seen that the precious metals are higher in the smartphones than the basic phones. Then we separated the printed circuit board, plastic and glass, and screen and other magnet and other parts from that. And we used only printed circuit boards for the treatment. We treated the printed circuit board with the solvent, which is sodium hydroxide, to remove the epoxy layer from the printed circuit board. And the metal content analysis was done using the AAS, atomic absorbance spectroscopy. And we checked the base metals, precious metals, and hazardous metals like copper, zinc, nickel, iron, aluminum, gold, silver, chromium, and lead. And after that, we checked the value of these all metals. We can see the pictures of the mobile PCB. In first figure, this is the phone I purchased, and I then separated this phone on the PCB type of mobile. Then pre-treatment was done using the 2 molar NaOH. We can see the removal of the epoxy layer, which is seen in a green color. This epoxy layer removal was essential as if the epoxy layer is present, it will inhibit the organism to treat with the metal content in the beneath of the epoxy layer. So epoxy layer removal was essential part. So I used 2 molar NaOH for the pre-treatment process. This NaOH could be reused for the 4 times. So the optimization of the NaOH was also done in my study. After that, the second step was the bioleaching of the base metal using iron oxidizing bacteria. For that, I have used leptospirillum dominated culture, which was I procured from the lab. This culture has efficiency to generate the ferric from the ferrous ion. It has oxidizing capacity, and the generated ferric has capacity to convert the zero valent metallic form of metals, which is present in the mobile PCBs, and convert it into the soluble form. So the mainly acidolysis process was done over here, and then I optimized the different parameters to extract the maximum copper from the PCBs. Here I have shown the different parameters which I have optimized. Process was optimized as 1-step and 2-step process. Cell process was optimized with cell and without cell. pH should be maintained or not maintained. Pretreatment is essential or not. Grinding is essential or not. Initial ferric concentration was also optimized. Initial pH, pulp density, how much amount of electronic waste could be treated, and the temperature, which is essential for the process. And we seen that 2-step process in which the organism is grown to produce the metabolite, and then the metabolite was used for the treatment process. We used with cell treatment process because without cell we seen the extraction was lesser. pH should be maintained. The pH should be maintained for whole the process. NaOH treated PCB was seen to give the higher extraction than untreated PCBs, as I have told that removal of epoxy layer is essential. In grinding process, pulverized, which is as a powder form, and unpulverized, in which we use the whole PCB. And we seen that whole PCBs is giving the higher copper extraction, because in pulverized PCB we seen the xerocyte formation is forming higher than in unpulverized PCBs, and xerocyte formation is decreasing the extraction. Ferric concentration was optimized at 18 grams per liter, 1.25 pH, 20% pulp density, and temperature we optimized to 50 degree for the copper extraction. And we seen that after the optimization process, around 9.8 fold of copper, 5.2 fold of nickel, 9.8 fold of zinc, 5.7 fold of chromium, and 3.6 fold of lead extraction was increased after the optimization process. Not only that, but the copper extraction time was reduced. Initially was 120 hours to 3 hours, and other metals it decreased from 96 hours to 3 hours. Also, we reached this process to 200 grams per liter of pulp density. So we can use this process for the scale up. After that, the fourth step was bioleaching of precious metals by cyanogenic bacteria. Cyanogenic bacteria has the capacity to produce HCN or hydrocyanic acid. This acid has capacity to solubilize the precious metals like gold and silver from the electronic waste. And first, second, and third process, which we already seen. The first was characterization, second was pre-treatment, and third was the biogenerated ferric treatment. For the bioleaching of precious metals by cyanogenic bacteria, we first isolate the cyanogenic bacteria from the rhizospheric soil. We adapt these bacteria to the electronic waste or mobile PCBs by sequentially transfer the electronic waste with the organism. After that, optimization of the initial pH and glycine concentration was done. pH is the essential part for the HCN production because HCN is produced only at the alkaline condition. So pH was optimized. Glycine is important parameter as glycine is the precursor for the HCN production. So glycine concentration was also optimized. So these both parameter was optimized for the highest HCN production. Then we optimize the parameter for the highest silver and gold extraction. For that, we optimize different treatment like 1-step, 2-step, and spent broth medium. And second was we optimize the pH and pulp density. Also, we use the untreated, NaOH treated, and bio-leach treated, which we already treated with the iron oxidizing bacteria, PCBs, for the maximum extraction of silver and gold. First of all, we can see the isolated HCN producing bacteria. Then we did the qualitative test for the detection of HCN production. Then we did the quantitative test for the HCN production. And finally, the highest HCN producing organism was screened out and selected for the further study. For that HCN production, we optimized the parameter pH and glycine concentration, and we seen that at 9.5 pH and 1% glycine concentration, highest HCN production was done. As I said, the process was also optimized for the highest extraction of silver and gold. So pH was optimized to 9.5. Process was optimized to 2-step process because we seen that at 1-step process and spent broth medium, the extraction of silver and gold was less. Treatment to the PCBs was also optimized. We used untreated, pre-treated, and ferric-treated PCBs, and we seen that when the copper was maximally extracted from the PCBs, the cyanogenic bacteria could easily extract the gold and silver, because if the copper is available in the PCBs, cyanogenic bacteria treat with the copper first and not to the gold or silver. So if the copper is already treated with the iron oxidizing bacteria, cyanogenic bacteria could easily treat silver and gold. So after the optimization, 6.4-fold of silver and 10-fold of gold extraction was increased. Not only that, the time duration was also decreased from the 8 days to 3 days. This process was done at only 250 ml flask level. So we did the lab scale study. First, we did 1 liter flask level study. After that, we did the 5 liter bioreactor study. And after that, we did the 25 liter bioreactor study. I have not shown here the 1 liter flask level data. Here we can see the pictures of 5 liter capacity bioreactor study, which was done with iron oxidizing bacteria. In first figure, we can see we have arranged the mobile PCBs in the bioreactor. This bioreactor was designed at the lab, and then we treated these mobile PCBs with 2 molar NaOH. And this we can see in the third figure that pre-treated PCBs has seen the removal of epoxy layer. Hello.
I request everybody to please keep mute.
Yes.
Sorry, Doctor, continue.
Okay. These pre-treated PCBs was then treated with the biogenerated ferric lixiviant, which we already produced with the iron oxidizing bacteria. And finally, we can see in the final figure the removal of all the metals, mainly copper, from the PCBs. In this bioreactor study, 1.1 kg of unpulverized MPCBs was used, and we treated this PCB and get around 293.9 gram of copper. After that, 25-liter capacity of bioreactor study was done. First figure, the washing step is done. Second figure, pre-treatment with the NOH was done. In third, we can see the biogenerated ferric treatment of the PCBs is shown. This study was done in two steps, two cycles. In first cycle was run for the 0 to 9 hours, in which we used 16.8 grams per liter of ferric, and we extracted the copper around 54.6%. In second cycle, which was run from 9 to 18 hours, for that 15.8 grams per liter of ferric was used, and we extracted the copper 33.4%. So around 90% of copper was extracted in the 25-liter capacity of bioreactor. 4.45 kg of unpulverized PCBs was treated in this reactor, and we got 910.8 grams of copper. After that, cyanogenic bacteria bioreactor study was also done at 2.2 liter capacity. The optimized parameter was run with this bioreactor, and we produced 16.6 millimolar of HCN. And this 16.6 molar of HCN was treated with the ferric treated PCBs. And after 72 hours of treatment, we got 37% of gold and 20% of silver. So from the 18 grams of ferric treated PCBs, we achieved 1.926 mg of gold and 1.548 mg of silver, which is similar to the 107 mg of gold and 86 mg of silver from 1 kg of ferric treated PCBs. After the recovery of copper was done, this is the picture of the leachate in which the solubilized metals is the present, and we did the physicochemical parameters of this leachate. After that, the cementation method was used for the recovery of copper. For the cementation method, we used iron nails. So first we treated the leachate with a little bit acid to decrease the pH, and then we added the iron nails to this, and cementation was done. And we can see in the third figure that copper precipitate we gained after the treatment, and this copper precipitate then solubilized, and in final picture we can see the copper sulfate crystals which we got after the whole cementation method. To confirm the study, we did the same EDEX analysis of all the samples. In first figure, we can see the original sample in which we can see the different peaks of different metals. In second figure, biogenerated ferric treated PCBs, same EDEX was done. And in third figure, we can see the cyanide treated PCB powders, same EDEX analysis. In first figure, there is lots of different peaks of different metals. In second figure, we can see that the copper and aluminum and these metals are removed from the second, which is ferric treated PCB. And in third figure, we can see there's this gold and lead peaks are removed over here. In fourth figure, this is the same EDEX analysis of the copper crystal. We can see the copper peaks only, and also there is impurities of iron only, not on other metals are not present in the copper hydrated crystals. So finally, this is the summary of my whole work. As I said, this process was the closed loop. First, we purchased mobile PCBs, then we washed it and air dried it. After that, alkali treatment was done to remove the epoxy layer. Epoxy layer is essential to remove, to treat the iron oxidizing bacteria to the metals. Then these pre-treated PCBs was treated with the ferric, which we already generated by iron oxidizing bacteria. These pre-treated PCBs was treated with ferric, and we can see the metals are solubilized in the leachate. These leaches was used for the cementation process, and finally after the cementation process, copper was uh, uh, copper as a copper sulfate formation we can gain, and it can reuse and it can be delivered to the market. These the PCBs which remained after the ferric treatment would be used for the HCN producing organisms treatment, and gold could be extracted and recycled. And after all this process, the finally ferric treated could be regenerated to, because in the process of ferric treatment, ferric is converted to ferrous, and this ferrous could be reused for the iron oxidizing bacteria to generate ferric. So this whole process is in a closed loop process. I would like to acknowledge Department of Biotechnology for the funding of my work, my guide, Dr. Devani R. Tipre. Special thanks to Dave sir for the guidance, Meenu ma'am, all the teaching and non-teaching staff, Department of Microbiology, my colleagues, and my family. And thank you so much. And please reuse, recycle the e-waste.
Thank you, Dr. Shital. Thank
Yes.
you. So indeed, this is a very nice presentation, and of course, a very nice topic. Very much.
Thank you, sir.
If anybody has any questions, please, just ask, or just you can type in chat box.
Yes, Jay, I want to comment on this presentation.
Sure.
Really, I'm appreciating Shital.
Thank you, sir.
The reason is that the electronic waste is a major problem in India,
Yes, sir.
and in the future, it will become more and more severe.
Yes, sir.
Who is not muted, please, I will remove otherwise. Please mute, everyone take the precaution. So, I'm very happy that you are presenting work today, and the way sir is present today in the discussion, Tipre ma'am is present, but in addition to that, the senior teachers like Bharat Shinde sir from Baramati, Ajay Srivastav sir from Jharkhand, and other senior teachers are also present. And let me tell you, I have also guided one student on the same topic.
Okay.
And he is present today, Dr. Nitin Adapure.
Okay.
He has also taken, separated copper, gold, and whatsoever you have told by the use of chemolithotrophic microorganism. And he also prepared the beads from that, and tried to prepare a gel and beads, and to use a technique for more separation. So he is also present today. Then Ambika Pathi is present, and many other seniors are present today. So I appreciate you. Keep this enthusiasm even in the future.
Yes, sir.
Microbiologist Society will always support you. You will receive a memento and certificate very soon.
Yes, sir. Thank you, sir. Thank you so much.
And now this topic is open for discussion to all. Please, you can ask the questions. There are comments. Nilima Pendarkar has given a comment, and many others have given a comment, but no question.
Yes, sir.
So, Dave sir also has given very good comment. But any question, please, Nitin, Dr. Nitin, I would like to make you comment, because you have done the work on the same topic, same title, everything similar. So please, Nitin,
yes,
I request you. Yes.
Yeah. So actually in between I have joined this presentation. So really a nice presentation, as sir said. And
Okay. Thank you, sir.
while seeing this presentation, I was like recalling my all data or all my work, my all
Yes.
PhD work and the work which I have carried out later as well. So
Hmm.
really a nice one, a good one. And I just want to mention that the candidate has not only studied the laboratory scale, but also the pilot scale studies as well.
Yes,
So,
sir.
so that is also a good thing. And uh, we have also actually set up like 100-liter plants over here about this processing of e-waste over here. And so I know the hurdles which are generally faced by
Hmm.
for carrying out such kind of studies over here. So really a good one. And the part which we haven't carried out is the HCN producing organisms.
Yes.
Like we haven't used the HCN producing organisms. So that is also a good thing that you have added it to your studies.
Hmm.
And because generally what happens is we can leach out the metals like copper, zinc, nickel, and tin by using chemolithotrophs. But the chemolithotrophs are not
Not able to extract.
they are not able to extract the precious metals particularly.
Yes,
So for
sir.
that you have used this particular strategy, and so you have scaled up that as well, I think, right?
Yes, sir. But only around 30 and 20% of gold and silver was extracted. So maybe some more optimization or more
Correct.
work could be done for the highest gold and
Yeah, sure,
silver
but you
extraction.
have started actually, and so
Yes, yes,
obviously
yes.
once you have started, so at certain time you will be able to make it out. And
Yes, sir.
like one more point I just want to mention ki, I might have missed that point. Have you extracted the precious metals like as you have extracted as you have recovered the
Recovered, yes.
copper?
No,
And what about the recovery of zinc and other metals, or even about the precious metals? So I might have missed that. So
sir,
can you
we
please
were
tell me?
thinking,
Yeah.
no, sir. Only copper recovery was done. We
Okay.
have thought about the recovery of different metals by electrowinning method.
Acha,
But
acha,
due
acha.
to COVID situation, it was not possible for me to
Mhm.
carry out my work.
Acha, acha, acha. Okay, 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 okay. Fine.
Yes.
But surely you will
Again,
get it. Yeah. Yeah.
yeah, I would like to tell Shital,
Yes,
Dr.
sir.
Nitin has a very good publications in a reputed journals.
Yes, sir.
So you can exchange now your research papers with Dr. Nitin, and you see that is a benefit today by this
Yes, sir. I will be in
activity.
touch with him.
Correct. Means if two scientists working on the same line,
Hmm.
exchanging their research to each other, that is a development of India, I consider.
Yes.
And
Yeah.
that is the biggest achievement today.
I'm
Correct?
sure I will welcome Dr. Shital as well as all people who are here in this meeting. I will request all of you people to visit our lab, so that we have established one 10-liter and 100-liter plant for this e-waste treatment with the support of PIRAC. So all of you are welcome to.
Yes.
Yeah,
He is
yeah.
in
Thank you, sir.
Aurangabad, Maharashtra, and his lab is there. I will share later on the contact number of Dr. Nitin.
Yes, yes, sure,
Yes,
sir.
sir. Sure.
Anyone can ask me on my WhatsApp. Okay. Thank you. Let
Okay.
us go, Jay. Now let us go to the second speaker. Is might be waiting.
Sure.
Thank you,
Thank you,
sir.
Shital. And I really would like to take this opportunity to thank Dr. Arvind Deshmukh for getting us all here. Sir, I would ask that Mannat is her name. Please also give her co-host, Mannat.
Yes, sir.
Hello, Dr. Anu.
Good evening, sir.
Good evening. So everybody, I would like to introduce our second speaker, Dr. Anu Kumari.
Good evening, all of you. First of all, I would like to thank all the organizing members of the
Anu, just a
microbiology.
minute.
Yes, sir.
Thank you. Dr. Kumari has done her PhD from MDU Rohtak, Haryana, and she has done her MSc from Department of Microbiology from the same university. She has done her BSc from Rohtak University. Dr. Anu has been associated with organizations like Association of Microbiologists of India, and she has a fellowship from Haryana State Council for Science and Technology. Dr. Anu has published more than a dozen research papers in national and international journals, and she has also participated in national and international poster presentations, seminars, and conferences. Now, I would like to welcome Dr. Anu. You may please start.
Thank you so much, sir. Good evening, all of you. First of all, I would like to thank all the organizing member of Microbiology Society India for giving me such opportunity to present my PhD work here. I am Dr. Anu. I have completed PhD Microbiology from Maharshi Dyanand University, Rohtak, Haryana. During my PhD, I basically worked on enzymology and biofuels production. And the
Can you share your PPT? Yes. Yes.
no, sir.
Yes, your screen is visible. You just have to open your PPT now.
No, PPT is open, but it is at the corner.
No, sir. This is not my PPT actually. This is
Okay.
Dr. Shital PPT actually.
Okay, okay. So Shital
Yes,
has
sir.
not stopped her PPT. Shital,
I
please.
think.
No,
I think
sir,
so.
I have stopped my screen.
Yes. Dr. Sital has stopped.
Okay, okay. So it's
Dr. Anu, please just stop your screen sharing and start it again.
stop screen sharing. And again, there may be some technical problem.
Yes, sir. Just a minute.
Sure, no worries.
Okay, sir.
First, you open your PPT on screen.
Yes, sir. I have already opened my PPT actually.
Then you go in sharing. Whether you are a co-host, yes, you are a co-host. No problem.
Yes, sir.
So you go on sharing option. Is there sharing option?
No, sir. Actually, the PPT of Dr. Shital is appearing on my screen.
Okay, so what
Yes,
I will
sir.
suggest again? What I will suggest?
Sir, I think I should
Uh,
start.
Can you mail your PPT?
Yes, sir.
Because I can share it within no minutes. You can start speaking within 2 minutes. I will share your PPT. So you mail
Okay, sir.
on my mail. The mail is Bio Society or sorry, AM Deshmukh. You take the mail AM Deshmukh, DESHMUKH, AM Deshmukh 1. Can you mail it?
Yes, sir. I am just mailing.
AM Deshmukh 1 @redipmail.com. @redipmail.com. Yes, I have not received up to the time. Should I write mail in chat box?
Yes, sir. Please write your mail. I think
Yes.
there is some.
Okay, I will write down in chat box. Yes, just you see, AM Deshmukh 1 @
Sir,
ready.
please check my PPT. I think now my PPT is visible. I think.
Yes, yes, it is.
Yes.
It is there. It is there. Okay, no
Yes,
problem.
sir.
Go ahead.
Okay, sir. Sorry for the inconvenience, sir.
No
Okay.
worries.
Yes, you can go ahead.
Okay, sir. The topic I'm going to discuss today is potential of lignocellulosic biomass for bioethanol production. Actually, this topic has become a topic of concern all over the world. As we all are aware that on the occasion of World Environment Day in the last year, the Honorable Chief Minister also released the roadmap for ethanol blending. According to this roadmap, the 20% ethanol blending is within reach in India by 2025. And if this happens, it would be a great achievement actually. And this topic is highly related to this problem actually. As we all know, approximately 84% of the world energy requirement is fulfilled by the non-renewable fossil fuels that are not only costly, but also have adverse effect on the environment as well as flora and fauna. So this overconsumption compels us to consider some sustainable sources of energy. In this view, lignocellulosic biomass like agriculture residues, for example, rice straw, wheat bran, rice husk, they are gaining a remarkable attention due to their renewable nature. Due to their easily availability in huge quantity, these feedstock have been considered as a feasible source for the production of various biofuels. During my study, I utilized rice straw as a substrate actually. Rice straw is why I consider rice straw? Rice straw is a rich source of lignocellulosic components. And if we utilize these agriculture residues as substrate, they don't only provide a suitable substrate, but they also solve some problems like open burning, some improper practices like open burning actually. And we all are aware about the pollution situation of the Delhi last year. And it was found that the stubborn burning was actually the only reason for the that problem. But due to recalcitrant structure of the lignocellulosic biomass, or we can say that the arrangement of the lignocellulosic constituents like cellulose, hemicellulose, or lignin, they make a barrier in utilizing these biomasses for various value-added products. So there is a need of ideal pre-treatment that can easily disrupt the various inter or intramolecular linkages of these biomass and make them easily accessible for converting them into various value-added products. And finally, the resultant reducing sugars can easily be fermented into various biofuels that can be bioethanol, acetoin, and so on. Keeping these points in mind, my PhD objectives were actually the production of cellulases by bacterial culture, that is Bacillus subtilis, subspecies subtilis, JJPS 300, and the fungal culture, that is Mycelioptera thermophila, BZTLR M23 in solid state fermentation. And then optimization of the pre-treatment condition by physical, chemical, and biological method. Further, the saccharification of the pre-treated rice straw using microbial cellulases. And then finally, the fermentation of sugar hydrolysate to bioethanol. This is the overview of the work done that can be considered for most of the biofuels actually. In the first step, usually the production of enzymes, that is cellulase in my case. In the first step, production of enzymes was carried out in solid state fermentation. And then in the second step, pre-treatment of the biomass was carried out by different methods like physical methods, chemical, and biological methods. Physical methods usually include steep pre-treatment, size reduction, microwave, ultrasonication, grinding, and so on. Similarly, chemical methods usually include acid and alkaline pretreatment methods. And the biological methods that usually include bacterial pretreatment, fungal pretreatment, and lacase pretreatment. And after this process, then the saccharification of this pre-treated biomass was carried out by the microbial enzymes that in my case was the microbial celluloses. And finally, the fermentation of the sugar hydrolysate into bioethanol was carried out by using fermenting microbes, that is Saccharomyces cerevisiae. In the first step, as I discussed initially, the production of enzymes was carried out by the bacterial and fungal culture. Initially, for the enzyme production, every factor has an important role on the microbial growth as well as on the enzyme production. Firstly, I optimize the enzyme production by bacterial culture using one variable at a time approach. In one variable at a time approach, I consider one factor at a time, that may be temperature, pH, carbon, and nitrogen source. During optimization, as I said, every factor has an important role. For example, if we consider about the temperature, temperature is an important factor in the enzyme production because it highly modifies the physical properties of the microbial membrane, as well as also stimulate the metabolite formed by the microorganisms also. Similarly, moisture ratio also plays an important role. For example, if the moisture ratio is so high than the optimum level, then it limits the oxygen transfer as well as the transfer of nutrient among the microorganisms. Similarly, if the moisture ratio is so less than the optimum, then it creates water stress conditions, or we can say that usually results in less delignification. Then the optimization, after optimization by one variable at a time approach by using the bacterial culture, it resulted in moderate production of cellulase, that is 9.70 unit per gram substrate. Similarly, the production of cellulases by fungal culture in solid state fermentation using one variable at a time approach resulted in good production of the cellulase, that is 56.06 unit per gram DMR. After one variable at a time approach, further the optimization of enzyme production was carried out by the statistical optimization, statistical approaches actually. That I consider placket-Vermeer design followed by the RSM, that is response methodology. Placket-Vermeer design usually was generally used to actually select the crucial medium component for the enhanced enzyme production. I consider 11 components in the placket-Vermeer design that were rhizotope, pH, moisture ratio, magnesium sulfate, moistening medium, sucrose, ammonium nitrate, PAGs, and twin 20, dipotassium hydrogen phosphate, and incubation period. And after placket-Vermeer design, it was found that the three factors highly enhance the enzyme production, and these three factors were twin 20, a surfactant, dipotassium hydrogen phosphate, phosphorus source, and the incubation time. Then these three selected factors were studied at 5 different levels, range from minus alpha to plus alpha, and range from low to high values. And these three selected variables, and then using RSM, 20 experiments were planned using the statistical software, keeping the three selected factors in mind, that are twin 20, dipotassium hydrogen phosphate, and incubation period. After statistical optimization, it was observed that there was more than 4.33-fold improvement in enzyme production, that is cellulase production, was achieved. These are the three-dimensional graphs showing the effect and interactions of the three selected factors that are between the dipotassium hydrogen phosphate and twin 20, and second is the incubation period and twin 20, and third is the between the incubation period and dipotassium hydrogen phosphate on the cellulase production. It was found that the dipotassium hydrogen phosphate up to 0.56%. and twin 20 up to 0.5%, and incubation after the incubation period of 5 days usually enhance the cellulase production, after which decline in enzyme production was observed. Then after the enzyme production, the second step is pre-treatment. Pre-treatment is usually carried out by the that is physical pretreatment methods, chemical pretreatment methods, and biological pretreatment methods. Physical pretreatments are usually considered as a mandatory pretreatment. It usually decreases the particle size so as to increase the available substrate surface area for enzymatic attack. But this pretreatment individually is not preferred, or we can say this pretreatment is usually considered along with other pretreatments. During my study, I considered different physical pretreatment methods. That was steam pretreatment, size reduction methods, microwave pretreatment, and ultrasonication period. Among all the physical pretreatment methods used, steam pretreatment resulted in maximum liberation of reducing sugars. Then after physical pretreatment methods, chemical methods were also used. Chemical methods usually include acidic and alkaline pretreatment actually. It was usually found that the chemical pretreatment using acidic methods usually gave good results in terms of liberated sugars. But it was found that the acidic pretreatment methods have many disadvantages actually as comparison to alkaline pretreatment. For example, some chemicals are very harmful for the environment, and secondly, some chemicals are very difficult to handle, like ionic liquids. And in the same way, some chemicals like dilute sulfuric acid, there is a need of corrosive resistant equipment for them. As compared to acidic pretreatment, pupils or researchers are favored alkaline pretreatment nowadays as compared to acidic pretreatment. As alkaline pretreatment usually disrupt the ester or glycosidic linkages of the lignocellulosic constituent, releasing in more liberated sugars. During my study, I consider different alkaline pretreatment methods like sodium carbonate methods, calcium carbonate method, and ammonia. And similarly, the acidic pretreatment was carried out by different concentration of sulfuric acid, citric acid, and lactic acid. Among all the chemical pretreatment used, the alkaline pretreatment, that is ammonia, at 10% resulted in maximum liberation of reducing sugar. So this selected pretreatment is further optimized by the statistical optimization, that is RSM, response surface methodology. This statistical optimization, that is response surface methodology, it is actually a multivariable statistical technique generally used to investigate the interaction between the selected component of the respective process. During the statistical optimization, it was found that more than 2.4 increment in reducing sugars was observed. And these are the three-dimensional and counter plot graphs showing the effect and interaction of ammonia and substrate concentration. And second is the time and substrate concentration. And third is the time and ammonia concentration. And results indicated that the enhanced in the saccharification at the concentration of 12% ammonia, 5% substrate concentration, and after the autoclaving time of 30 minutes. After the statistical optimization, the scale-up of saccharification of the selected, that is ammonia pretreated rice straw, was carried out for 48 hours. With varied amount of substrate.
बज रहा है उससे भी तैयारी होता है.
Excuse me, everybody. Please keep yourself on mute.
Then after this statistical optimization of saccharification, then the scale up of the saccharification of ammonia pretreated rice straw was carried out with varied amount of substrate, varied from 0.5 gram to 25 gram. Result indicated the liberation of almost similar amount of reducing sugar, or we can say a sustainable amount of sugars were liberated. But as compared to physical and chemical pretreatment, biological pretreatment is highly recommended pretreatment actually, because it utilizes the microbial ability to degrade biomass, or we can say for enzyme production or for various other biodegradation process. During my study, biological pretreatment was carried out by both the bacterial as well as the fungal culture. For biological pretreatment, the biomass was taken at different time interval, and then the percentage loss of biomass, and then production of cellulase, and finally the sacrification of the pre-treated biomass was carried out. In case of bacterial culture, it was found that the maximum percentage loss of biomass, that is 25.88%, was achieved in 10-day-old bacterial pre-treated biomass. And then the percentage, whereas the sacrification of the sacrification of the 6-day-old pre-treated biomass resulted in maximum sacrification, that is 49.24 mg per gram, reducing sugars. Whereas in case of the fungal culture, both the percentage loss of biomass as well as the production of maximum cellulotic enzymes both occurred in 9-day-old pre-treated biomass. Whereas, however, the sacrification of 5-day-old pre-treated biomass, fungal pre-treated biomass, resulted in maximum liberation of sugar. After that, the biological pretreatment using the lactase of Ganoderma lucidium was also carried out. Initially, I optimized the pretreatment of rice straw by taking different unit of lactase that varied from 250 units to 1000 units. But result indicated that almost similar amount of reducing sugars were liberated with all the units used. So I selected 250 units of lactase for further pretreatment process. After that, the sacrification of the lactase pretreated biomass was carried out at different time intervals, and it was found that maximum liberation of reducing sugars occurred. Maximum amount of reducing sugar, that is 162.82 mg per gram, were liberated after 72 hours. Further, simultaneously lactase pretreatment and sacrification of rice straw was carried out at different time intervals, and it was found that a further increase in liberated sugar, that is approximately 182.82 mg per gram substrate, was liberated after this pretreatment. Then after the pretreatment, the third step is the sacrification of the pretreated biomass by using bacterial as well as the fungal cellulases. And firstly, I optimized the sacrification by different factors like enzyme dose, pH, temperature, and as well as the incubation time. Enzyme dose is found to be an important factor in sacrification. It was usually found that with the increase in enzyme dose, usually increase the interaction between the substrate and enzyme, that finally resulting into releasing of more reducing sugars. During my study, it was found that the sacrification of ammonia pretreated rice straw result by the fungal cellulases resulted in maximum liberation of reducing sugar, that is 369.98 mg per gram substrate. But this enzyme unitage is somewhat so high, so I selected 20 units as an optimum enzyme dose for carrying out further experiments or further sacrification experiments. Then the effect of different pH on the sacrification process was also investigated. pH plays a crucial role in sacrification. Because it plays an important role in forming of a relevant substrate and enzyme complex, or we can say it also prevents the unproductive binding of enzyme on the substrate. So it is an important factor in sacrification. Among all the pH used, it was found that maximum amount of sugars were liberated at pH 5, that is 246.33 mg per gram substrate. Then among the different temperature used, maximum amount of sugars were liberated at 60 degrees Celsius, and this 60 degrees Celsius is actually also the optimum temperature for fungal culture also. Then finally, the effect of incubation time on the sacrification was also investigated. It was found that there was gradually increase in liberated sugar was observed up to 24 hours only. After the 24 hours, there is decline in reducing sugars occur. This might be due to the inhibitory effect of the produced sugars on the enzyme or substrate. Then after the pre-treatment, biophysical characterization of the biomass, that is both untreated and pre-treated biomass, was carried out by the different techniques. Initially, the estimation of the cellulose, hemicellulose, and lignin in both the untreated and pre-treated isotope was carried out by using Zing and Zhang method. And it was found that after the pre-treatment, there is increase in cellulose and hemicellulose content, but there is decrease in lignin content from 25 to 40.9%. That indicated that this pre-treatment has a, we can say this pre-treatment gave better results or good results. After this, the biophysical characterization was carried out by the different techniques that are FTIR, for infrared spectroscopy, and then XRD, and then scanning electron microscopy. FTR is actually analytical technique used to calculate the compositional as well as the structural differences in the biomass after pre-treatment. FTR spectra usually show the difference in the intensity of untreated and pre-treated biomass. As we all know, the reason between the, in the FTR spectra, the reason between the 3200 to 3400 and the reason between the 2800 to 3000, it has previously assigned to CH and OH stretching of lignin. Results clearly indicated the reduction in these peaks in pre-treated biomass. That clearly indicated the, we can say that the degradation in lignin, or we can say that the degradation in lignin, or we can say that the some modification in the cellulose after pre-treatment. Similarly, after FTR, then XRD, this is the XRD spectra. And this is the XRD spectra. XRD spectra also clearly indicated the differences in the XRD peak of both untreated and pre-treated biomass. It was generally found that the peaks around the 16 degree and 22 degree, these peaks usually indicated the crystalline and amorphous form of cellulose. As we all know, crystalline is highly resistant to any pre-treatment or enzymatic attack. So in the results clearly indicated the elevation in peak in the pre-treated biomass, or we can say that the presence of more amorphous form in the pre-treated biomass. That also show a positive, if we can say that the pre-treatment also showed a positive effect on the biomass. Then after this, scanning SAM technique was used to analyze the morphological differences in the untreated and pre-treated biomass. As it is clearly indicated in the figures that untreated biomass usually show rough, regular, and smooth structure. Whereas in case of pre-treated biomass show some irregular or scattered structures. That might be due to the effect of pre-treatment, due to which there is this, we can say disruption in cellulosic fibers might be occur. And then after the biophysical characterization, the analysis of reducing sugars was carried out by using thin layer chromatography as well as HPLC, that is high pressure liquid chromatography. And in this figure, lane 1 and lane 2 usually represent the standards, that are glucose and cellobiomes. And lane 3 to 7 represent the various hydrolysis products taken at different time interval after the saccharification. It was found that okay, sir, I just require 5 minutes. Okay. And the results clearly indicated that the presence of various intermediate sugars along with some monomers, that is glucose, and oligomers, that is cellobiomes. Then HPLC profile of the enzymatic hydrolysis of ammonia treated rice clearly indicated the presence of high amount of monomeric and oligomeric sugars as compared to the untreated biomass. And then finally, the fermentation of the enzymatic hydrolysis of the ammonia pretreated rice was carried out by Saccharomyces cerevisiae at different factors like time interval, pH, agitation speed, temperature, and hydrolyzed sugars that finally resulted into 24.37 gram per liter bioethanol. And then summary and conclusion. Then optimization of the cellulase production by bacterial and fungal culture resulted in enhanced cellulase production after optimization. Among all the pretreatment used, alkaline pretreatment, that is ammonia, resulted in liberation of high amount of reducing sugars. Then FTR, XRD, and SAM analysis clearly revealed the disruption in cellulose or hemicellulose, as well as the a suitable, or we can say that a significant delignification. After that, the analysis of reducing sugar by TLC and HPLC confirmed the presence of various monomeric and oligomeric sugars. Finally, the fermentation of the enzymatic hydrolysis by Saccharomyces cerevisiae enhanced the ethanol production after optimization. After this study, a cost-effective and eco-friendly process for cellulase production was developed for enhanced saccharification, followed by fermentation into bioethanol. The significant increment in cellulase production, liberated sugars, and bioethanol production was achieved in this study. However, a large-scale study is still required for the industrial application of this process. That's all about the study. Thank you so much.
Thank you, Dr. Anu. Thank you very much. It was very nice presentation.
Very good, very good, very nice topic. That, as you have told, rightly told, it's a burning and hot cake in India.
Yes, sir.
The reason is that Honorable Gadkari ji, Transport Minister of India, he is always making an appeal that we want to use ethanol as a biofuel.
Yes.
But there is a scarcity of ethanol in India. Your topic is the same. That is, we want to enhance and optimize the production of ethanol from raw organic material.
Yes, sir.
And you have done the best research. Who was the, by the way, what is the name of your guide?
Dr. Vijender Singh. He is designed as a professor in the Central University of Haryana, Mahendragarh.
Okay, very good. Convey my congratulations to him also.
Thank you, sir.
And excellent work. Now, many other teachers are there. Deepak Sharma is also there. Senior scientists are there. Anyone want to ask the question? He is free to ask the question. If not, I request you, please make any comment on that. Because Vivek Parab is there. In addition to that, Ambika Pathi, Tipre ma'am is there. So you can ask, Dave sir is also there. Surekha Patil ma'am is there. Pendarkar ma'am is there. So either ask the question or make any comment. Please. Sharma sir. Hello.
Yes, sir. Yes, sir.
Yes.
It was a very nice presentation, sir.
Yes.
Good work done by the girl, and she has presented very well. All the techniques are used classically, and this right now many people are using them, and the work is also good. And she is getting very good amount of enzyme, around 56 units per liter per gram of dry biomass. I think it is a good one.
Correct.
So,
Correct.
it could be commented.
Tipre ma'am. Hey, sorry. Continue, sir. Continue.
Yes, sir. Because the work is I can say that it is a need of the time, because a lot of pollution and the problem is created by the rice straws. So it could be a very helpful
Yes.
thing in to remove this pollution, sir. I think.
Yes. That's why such type of work should be encouraged, and they are being encouraged by the government.
Yes, sir. Yes, sir. Definitely.
Thank you. Tipre ma'am, please. Dave sir, please make a comment. Dave sir. Bharat Shinde sir. Dr. Shinde. Okay. It's all right. Dr. Vivek, you want to make any comment, Dr. Vivek? He has written the comment in chat box. Dr. Vivek.
Okay, his mic is not working, so I would just read out his comment.
Okay.
Dr. Vivek is saying, yeah, sir.
Ambika Pathi. Ambika Pathi. Okay, it's all right. So, Jay, you can go ahead.
Thank you. So thank you to Dr. Kumari and thank you to Dr. Shital Thacker. And thank you everybody for being here. It was a pleasure.
Thank you so much, sir.
All right, and if anybody have any question, please write in the chat box or later on you can send it.
There is one question.
Okay.
Tipre ma'am has written the question.
Okay, I would just read out to you. What is the cost calculation of bioethanol production in your study?
Sir, actually, I didn't calculate the cost of bioethanol production, but I calculate the cost of enzyme production. It is approximately 45 USD.
45
USD.
Per
gallon or what?
Yes, sir.
Okay.
So, as Tipre ma'am has asked the question, it is very correct. Because whenever you are doing any investigation, the last question is what is a bioeconomics?
Yes, sir.
And you can continue on that. And the Dr. Vivek has written that it is a quite cost effective. So you see again, bioeconomic is a very important.
Yes, sir.
So you can do the work further. That is a good suggestion. You continue your research.
Sure.
And find out the bioeconomics, whether it is affordable or not.
Okay, sir.
Correct?
I will definitely do that.
Yes. Any other question? You can type. It's a very good questions. And you see, you got one suggestion. That is a benefit. That is achievement of today's seminar. We never expect 100% achievement. 1% achievement is also called as achievement. 2% achievement is also called as achievement. So I am very happy today. Both the speakers were very enthusiastic, young, dynamic. You are ample scope in your life in the future. Continue your research and keep in touch with microbiologists to society. I am making appeal to you as a president of Microbiologist Society. Thank you, Jay. You can continue.
Thank you, Dr. Deshmukh. I would just like to say Dr. Meenal Trivedi, our Gujarat State President, has conveyed her wishes and she has thanked everybody who has participated in this webinar. And once again, I would like to thank both of our speakers and everybody who have participated. And I think that would be all. Let's meet up next Sunday.
Okay, thank you. And tomorrow you can go on YouTube channel of Young Bio Society. This will be available tomorrow. Okay.
Yes. Yes.
Oh.
Yes.
Thank you.
Okay. Goodbye, everybody.
Okay. Thank you.
Thank you.